Hello, everyone. Welcome to today's webinar. And I'm very glad to have you here with us. I will introduce myself. My name is Serling, and I am a project manager in the Pharmacology Center at GemPharmatech. And my role involves overseeing the development of animal models for studying metabolic disorders and their applications. And today, I'm very excited to share with you the latest development in our NASH models and its applications in preclinical study. Our team has dedicated a great deal of effort and resource toward refining our NASH models. So I'm very eager to share our data and participate in a productive discussion with you all here. Thank you for joining us today and for your attention. As we know, non-alcoholic fatty liver disease is the spectrum of chronic liver disease. It is characterized by an accumulation of fat in the liver, leading to inflammation and liver cell damage. Non-alcoholic steatohepatitis, which is NASH, is a more severe form of NAFLD that can lead to the development of cirrhosis, hepatocellular carcinoma, and increased mortality related to liver disease. So including obesity, high cholesterol, and type 2 diabetes are all potential high risk factors for developing NAFLD or NASH. And currently, about 25% of the global populations is estimated to have NAFLD. Of them, 25% of them developing NASH. So there is a very urgent to develop effective treatment for liver disease. And NASH has already emerged as the third most common reason for liver transplantation and the second leading cause of hepatocellular carcinoma. So, as we know, NASH in humans are characterized by the presence of steatosis, long-build inflammation, hepatocyte injury such as blooming, and fibers in the liver. At the same time, this disease accompanied by metabolic disorders such as obesity, hyperglycemia, hyperlipidemia, and insulin resistance. So in 2022, a new definition for NAFLD was proposed, known as metabolic associated fatty liver disease. So according to this new definition, the diagnosis of NAFLD requires evidence of hepatic steatosis along with one of the following criteria, including overweight or obesity, the presence of type 2 diabetes, or signs of metabolic dysregulation in patients who are very lean. With this definition, the diagnosis of NAFLD or MAFLD is based on the presence of metabolic dysfunctions in patients. With this new approach to diagnosis will have an impact on the development of animal models for NASH, as the metabolic characteristics of models will receive more attention. Even though the significant public health impact of NASH, the current treatment options for the disease are very limited. One of the primary reasons for this is lack of the relative animal models for NASH. While there are several dietary and genetic models for NASH available, however, very few replicate all the metabolic, histological, and genetic features of human disease. With these limitations have hindered the development of effective therapies for NASH. But fortunately, significant efforts are being made to improve animal models for NASH to better reflect the disease complexities. So in the current landscape of the preclinical animal models for NASH, there are several classic models, including diet-induced, toxin-based, Harvard diet-induced, and genetic modified models. Each model has its own advantages and limitations. As we know, Harvard diet and Western diet models are often preferred as they mimic the natural progression of NASH in humans. But these models typically produce only mild liver injury, including the steatosis, mild inflammation, mild blooming, and mild fibers in the liver. And this mice model has a slow progression of metabolic disruption, which limit their application in the drug efficacy study. So next is modified diet such as methionine and the choline deficient diet, and as well as the choline deficient hybrid diet. This diet has been developing to accelerate a severe NASH phenotype in mice. Another mice model is chemical like carbon tetraglyceride and streptozotocin has also been used in combined with hybrid diet to induce severe NASH phenotype. But even these special diets and the chemical-induced models replicate some histology changes, features of human disease in a very short time, but the lack of metabolic disorders makes them quite different from human NAFLD. So there is a need to develop NASH mouse models that have a relatively short time and is also accompanied by metabolic syndromes. So today, the first model I want to discuss in this presentation is the monogenic mutation models. This mice model was established by knockout the leptin receptor gene in BKS mice by CRISPR-Cas9 technology to generate these BKSDB mice. Leptin receptor gene was knocked out in these mice, so these mice have symptoms of type diabetes, including hyperglycemia, hyperinsulinemia as early as 4 weeks of age. Additional BKSDB mice exhibit increased liver enzyme increase, including the LTST levels at X week old, which indicating this mice model have liver damage. At 12 weeks old, significant hepatic steatosis, accumulation of fat in the liver, developed in BKSDB mice. This further indicating the progression of NASH in this model. So the BKSDB mice model is a valuable tool for studying the mechanism of metabolic disorders and assess the potential treatment for NASH and the related complications. So next, the Western diet was also shown to accelerate the development of NASH in BKSDB mice model. This, as shown in this data, after 4 weeks of Western diet feeding lead to the significant increase in the body weight, increased blood lipid, and the liver enzyme including LT levels compared to the control mice. Our histological data also show that steatosis and the lipid accumulation was induced after 4 weeks of Western diet feeding, as demonstrated by both H&E and oil staining. In addition, the NAFLD activity score was found to be significantly high because DB mice fed with a Western diet compared to those fed with a normal diet, indicating the most severe liver pathology changes in BKSDB mice fed with a Western diet. So this result suggests that BKSDB mice model is a useful tool for studying the pathogenesis of NASH and evaluating potential treatment for NASH. To establish a Western diet-induced NASH model, here male BKSDB mice at the 6 weeks old were fed with the diet for 4 weeks. After these mice being separated into several groups based on their plasma levels of liver enzyme LT. LT is a primary indicator of liver damage. During the 4 weeks Western diet feeding period, some mice were treated with MGR-396, and the efficacy of treatment was evaluated at the study endpoint. So here is the data of the positive drug treatment in BKSDB NASH models. By administration of MGR-396, significant attenuated NASH phenotype in BKSDB mice. Following treatment, there was a decrease in the liver size, plasma cholesterol, and the low-density lipid cholesterol and the liver enzyme, the enzyme LT levels. The histology data also show that MGR-396 treatment result in the reduction of steatosis and lipid accumulation. This result was demonstrated by both H&E and oil staining. Additional MGR-396 also lead to the improvement in the NAFLD, as shown by the reduction in the NAFLD activity score. So this data obtained from BKCB mice suggests that our developed NASH model can successfully mimic key characteristics of human NASH, such as the metabolic dysfunction, including obesity, hyperglycemia, hyperinsulinemia, and hyperlipidemia, as well as liver histology changes, including inflammation, fibrosis, and hepatic steatosis. These findings are highly encouraging and suggest that our model may be available in preclinical drug development. The second mice model is our B6-AMS1 NASH models. As we know, AMS1 encoding proteins that function the microtubule organization. Mutation in this gene are associated with Astrom syndrome. This is a rare genetic disorder. It's characterized by various complications, including vision and hearing loss, obesity, as well as insulin resistance and cardiomyopathy. Astrom syndrome types appear in infancy and presents a range of symptoms that progress over time, leading to significant health complications and even death in humans. So this condition is caused by mutation in the exon 18 and 16 of the AMS1 gene, which cause a frame shift, resulting in the insertion or deletion of the nucleotide. So in this B6-AMS1 deletion mice, an 11 base pair deletion in X8 causes a frame shift and premature termination coding with this domain, leading to the absence of functioning AMS1 protein. So these mice spontaneously develop obesity, diabetes, and fatty liver disease. As the data show, AMS1 mice fed with a child diet showed an increased body weight compared to the B6 control mice. Hyperglycemia in AMS1 mutant mice were also observed as early as 8 weeks old. Along with the hyperglycemia, AMS1 mutant mice exhibit impaired glucose intolerance compared with the control mice, indicating a disorder of the glucose metabolism. Also, AMS1 mutant mice show impaired glucose tolerance as early as 11 weeks old. AMS1 mutant mice also display liver injury. These mice had a higher plasma ALT and ST levels compared to the control mice from 16 weeks old. Control and the AMS1 mutant mice were sacrificed as 20 weeks old, so consistent with impaired liver function, the liver size is larger and the weight. The histology analysis showed that severe hepatic steatosis, lipid accumulation, and liver fibers in AMS1 mutant mice. So in conclusion, AMS1 mutant mice spontaneously develop the liver pathologies of NASH combined with metabolic disorders. This mice model at 6 weeks old has been used as a spontaneous NASH models. So in the efficacy study, these mice were divided into several groups based on the plasma levels of ALT, which is indicators for liver damage. Following 5 weeks of treatment with a test compound, the research can assess whether treatment effectively reduced ALT levels or improved liver histology changes. Here is the text articles in B6-AMS1 deletion mice. As we know, semaglutide is a GLP-1R agonist. It is developed for weight loss and blood glucose regulation. It has shown promising results in phase 3 clinical trials. So during the 5-week treatment period in AMS1 NASH mice, semaglutide was found to attenuate the body weight gain and post-standard blood glucose levels. Additional semaglutide treatment resulted in a significant decrease in the plasma cholesterol, high-density lipid cholesterol, low-density lipid cholesterol, as well as triglyceride levels. The treatment also led to a decrease in the liver enzyme ALT and ST levels, which is important histology changes also show a reduction in the liver lipid droplet in the semaglutide treated mice compared to the control mice. And the decreased steatosis score and liver lipid contents were observed by H and E staining results. Here is another two positive drugs, which is FGF21 analog and PZP drugs. These two drugs improve the NASH phenotype of B6 AMS1 deficient mice. As we know, FGF21 is a protein that is involved in the regulation of metabolism and energy balance in the body. It is produced in the liver and adipose tissue. So research on FGF21 has shown that it has potential as a therapeutic target for metabolic disorders such as obesity, type 2 diabetes, and fatty liver disease. Tirzepatide is another potential treatment for type 2 diabetes and obesity. It's a dual GIP and GLP receptor agonist. So in this B6 AMS1 deletion model, treatment with these two drugs result in a decrease in the body weight, while the food intake remain unaffected. Additional treatment with these two drugs result in a significant decrease in the levels of liver enzyme LT and ST levels, indicate improved liver function. Furthermore, the plasma triglyceride, cholesterol, high-dense lipid, low-dense lipid were decreased, suggesting the benefit effect on the lipid metabolism. The effect of these two drugs on steatosis, inflammation, blooming, and liver fibers in a preclinical model of NASH was evaluated using H&E and serial staining. So this result indicates the treatment with either of them significantly decreased both the NAFLD score and the liver fibers compared to the control group. These mice also can develop NASH by Western diet feed. To accelerate the development of NASH, these mice were fed with a Western diet. So with a very short period of just 4 weeks treatment, Western diet fed mutant mice showed a significant increase in the plasma cholesterol, low-dense lipid cholesterol, and the liver enzyme levels, including the LT and the ST levels compared to the B6, and as well as the mutant mice fed with a char diet. However, only a slight increase in the body weight were observed in the Western diet group compared to the char diet group. Here is a comparison between the young AMS1 mutant mice fed with a Western diet for 6 weeks and the older mice fed with char diet for 21 weeks. As the data show, these young mice fed with the Western diet and the older mice fed with the char diet had a similar metabolic disorder phenotypes, and the liver injury is also very similar. The liver pathology analysis based on the plasma chemical shows that the Western group have a large and pale livers. Hepatic steatosis, inflammation, and fibers were also observed in AMS1 mutant mice fed with a Western diet compared with the control mice. Furthermore, the NAFLD activity score in young AMS1 mutant mice fed with a Harvard diet was higher compared to the older mice fed with a char diet. So this study demonstrates that the AMS1 NASH models replicate most of features observed in classic NASH models, including metabolic disorder and the liver pathology changes. Important, the AMS1 NASH models can be established much faster, which is induced by Western diet, making it a very valuable preclinical research tool. Here is the efficacy study used the Western diet induced NASH models. So to induce NASH-like pathologies, male B6 AMS1 deficient mice aged at 6 weeks old were fed with Western diet for 6 weeks. So based on their plasma levels of LT, which served as a primary indicators for liver damage, the mice were divided into separate treatment groups. After several weeks of treatment with the text articles, the efficacy was the drug efficacy was evaluated. During the treatment period, MGR396 and the TCP in the Armstrong NASH models, the body weight was closely monitored. The data demonstrated that both drugs were effective in reducing the body weight gain, liver weight, as well as fat mass. The following data show that MGR396 show a significant effect on the plasma cholesterol, high-density lipid cholesterol, and low-density lipid cholesterol, as well as triglycerides in Armstrong mutant mice, which was consistent with the weight loss effect observed in the treatment group. Furthermore, in compared to the control mice, the evaluated plasma levels of liver enzyme, including ALT, AST levels, were significantly decreased after treatment, while both drugs demonstrate a strong effect on the plasma triglyceride. So this study aims to assess the effect of different concentration of MGR396 on hepatic steatosis and fibers in B6 ARMS1 deletion mice while fed with a Western diet. Here, steatosis and fibrosis levels were determined by H&E staining as well as cirrhosis staining. The results show that both treatments lead to a decrease in the liver weights of mice. And treated with high dose of MGR396 result in the significant reduction in the NAFLD score compared to the vehicle treatment mice. However, TZP treatment did not lead to a similar effect. Neither MGR396 nor TZP has a significant impact on the liver fibers levels in these mice. So in conclusion, the Armstrong mutant mice used in this study were found to spontaneously develop multiple metabolic dysfunctions, including obesity, hypoglycemia, glucose intolerance, dyslipidemia, and hepatic steatosis. And these features are consistent with the symptoms of human NASH. And very important, we found the fed mice with Western diet accelerated development of NASH phenotypes. So our results provide strong support for the use of Armstrong mutant mice as a preclinical model for evaluating treatment for NASH and other related metabolic disorders. So the final model discussed is the chemical-induced NASH models. This model is established in B6 mice fed with a Harvard diet or Western diet for several weeks, followed by twice-weekly treatment with carbon tetraglyceride. This method leads to the development of severe hepatic steatosis and liver fibers. INT-747 is a drug that's currently being researched for its potential use in treating liver disease like NASH. So it works by targeting specific receptors in the liver that are involved in regulating bile acid synthesis and fat metabolism. The effect of drug on steatosis, liver fibrosis, and lipid accumulation in a NASH model was evaluated by H&E staining and the cirrhosis staining, as well as oil staining. These results show the treatment with INT-747 lead to significant decrease of the both NAFLD activity score and the liver fibers score compared to the control group. This is another positive drug treatment in this mice model. This data show MGR396 can mitigate the hepatitis phenotype in Harvard diets and the chemical-induced NASH models, and the NASH scores and the fiber scores were significantly reduced. So last, I would like to take a moment to introduce the drug screen project at GemPharmatech. Our project aims to develop targeted specific drug to treat a range of metabolic disorders, including obesity, diabetes, dyslipidemia, and NASH. So to achieve this goal, we have created a series of humanized models for these disorders, which are available in our website. So if you are interested in learning about this project and these models, you can visit our website or reach out to us for further information. So that's all. Thank you for listening. Next, we will step in a question and answer session. If you have any question, you can type your question in our chat room. Thank you for listening. So now is the question, what kind of Western diet did you use in the induction of the Armstrong NASH models? For this mouse model, we use the rodent diet with 40% calorie fats with pale oil and with 2% cholesterol in the diet. You can search this diet in the research diet, and I can provide you with a diet ID. So there is another question. Ask us, do we do several types of work to help evaluate drug efficacy study, or you do several gene engineering mice models? So for this question, our company provide a one-stop solution, including the mice model generation. After the mice was generated, we can provide you with preclinical service. If you have drug to evaluate, we can help to do the efficacy study in our facility. So we can do mice generation, mice breeding, and the preclinical service. So for the slides, you can subscribe in the YouTube to follow GemPharmatech, and you can find this video in the YouTube. How about the hepatocellular carcinoma models? So for these models, some published paper use Western diet as well as the carbon cholesterol to induce HCC models, but it may take a very long time, about 24 weeks. So how about the change of food intake upon the treatment of MGL-396 and semaglutide? So for the food intake for MGL-396, we did not observe the significant difference between the treat group and the vehicle group. It means with the MGL-396, the food intake was not affected. Is unaffected. So for the semaglutide, we observed the semaglutide can decrease the food intake. If you are interested in our mice models, you can visit our website and type your gene you are interested in, or you can search animals you are interested in. If you can't search the mice model in our website, you can contact us and we provide a service for customer mice. That means we can customize for you. You're welcome. Do you have any other questions? You can feel free to ask us. And if I can't answer your question, we will contact you after this meeting. If you are interested in either of these models, I can send some information to you. Okay, we will stop here. If you have any other questions, please feel free to contact us or visit our website. We are very happy to provide any service, including the customized models, mice breeding, and the preclinical service. Thank you.